Hi everyone, my name is Sorenza. I'm a PhD candidate at the Krembil Center for Neuroinformatics in the Whole Brain Modeling Group. It is my pleasure to be presenting at the educational course at OHBM on Connectome-based neuromass modeling. So here is an overview figure of this type of modeling. And as you can see, different elements are assembled together to generate a whole brain model. And an important features are the nodes of the models describing the local dynamics. So my talk delves a bit more into the specifics of those local dynamics, which are represented by neuromass models. I will start the presentation with some background to give context on what we are modeling and why. I will then go over what is a neuromass model and their general features. And within the vast landscape of neuromass model, I will then focus on three specific models, each with a different particularity to kind of present what is possible with these models. And building upon the understanding of individual neuromass models, I will then introduce the concept of connectome-based neuromass model simulation and explain how these neuromasses then are integrated into a connectome framework to simulate whole brain activity. And then finally, I will go over a few applications of the model in a research and clinical context to illustrate their relevance in the field of neuroscience. Thanks to neuroimaging devices, we have been able to visualize and capture many structural and functional features of the brain, which allowed us to investigate cellular processes, brain dynamics, and so on. The modalities that record brain activity can be broadly divided into microscopic or macroscopic devices. The type of modeling we are seeing today aims to replicate whole brain activity, which is commonly measured with EEG, MEG, or fMRI. So they are all non-invasive recording devices. The EEG measures the neural electrical activity of millions of neurons over the entire scalp using electrodes attached to the surface. And similarly, MEG records not electrical activity, but the magnetic fields produced by electrical currents using highly sensitive sensors. And since EEG and MEG simultaneously captures the activity of all these neurons with a high temporal resolution, repetitive patterns emerge. These patterns are called neural oscillations, and they are important events in the brain that play a key role in a number of cognitive processes. And they are categorized into different frequency bands, and they are each associated with a specific state of activity. So for example, the alpha frequency band, which is a peak in the power spectrum around 10 hertz, is characterized by resting state. It is what you commonly see during resting state. And an fMRI detects changes in blood oxygenation to infer neural activity using a scanner with a large magnet. So compared to EEG and MEG, fMRI is an indirect measurement of neural activity. Essentially, it captures the changes in the level of oxygen in different brain regions to provide a measure of how the activity of these regions changes over time with great spatial resolution this time. And this is what is called the bold signal. And even though other modalities are also able to capture whole brain activity, such as FNIRS, PET, and so on, I focus your attention here on these specifically because the model we are going to see and also encounter in the hands-on session attempt to replicate phenomenon that can be observed with EEG, MEG, or fMRI. So why do we want to model this phenomenon? Well, this is because the underlying mechanisms of the observations of the whole brain scale, such as brain oscillation, they remain relatively unknown and they lack a clear definition. Therefore, in order to explore the intrinsic processes of the brain and bridge the gap between different scales, mathematical models of the brain are particularly useful. And one method is connectome-based neuromass modeling. And so it uses a neural mass model as a unit, which simulates neural activity at the mesoscopic scale, which is between microscopic and macroscopic brain. So it simulates the brain regions, and the different neural masses are then assembled together to represent whole brain activity. So what is a neural mass model? Well, neural mass models are a specific type of mathematical modeling approach that can capture the collective behavior of large groups of neurons and synapses using only a few equations. So within a cortical column, you have many neurons and many different types of neurons, such as pyramidal cells and interneurons. But rather than focusing on individual neurons, neuromass models aggregate neural populations with shared characteristics, such as excitatory or inhibitory properties, and then the equations describe the activity of this population as a whole. And so by simplifying the dynamics and omitting certain spatial information, neuromass models are still able to capture changes in firing rates within these neural populations. So obviously, one major advantage of neuromass models is the reduction in complexity by condensing the number of dimensions and equations that need to be integrated, while still gathering information about the dynamics of numerous ensembles and shed light on their behavior. However, the reduction in complexity comes at the cost of keeping biological details. So the objective here is kind of to strike a balance between mathematical tractability and biological plausibility with these neural mass models. So they provide a powerful tool for examining the coarse-grained activity of large neural populations, enabling us to really hone in on those larger scale brain dynamics. And through the simplification of the equation, then we have a model that is mathematically manageable while having a biological foundation. So within neuromass models, different modeling approaches can be used, including convolution versus conductance-based models. For conductance-based models, very high coherence between the neurons is assumed to the extent that the dynamics of neuron populations resembles the dynamics of each single neuron. So the mathematical equations then follow the same structures as single neuron conductance-based models, such as you might be familiar with the Hodgkin-Huxley model. And since distinct types of ionic currents are explicitly modeled, a direct relationship between model synaptic processes and physiological mechanism can be determined. In contrast, convolution-based neuromass models, they rely on empirical observations of the collective response of neural population to inputs, and then a model is built that will capture the system's response to that input. And this is to take into account the fact that complex systems may exhibit emergent behavior that cannot be explained solely by the behavior of individual components, but rather arises from the collective interactions and relationships among them. And thus the rules governing the behavior of a complex system may differ from those at a lower level of organization, as the system as a whole can be more than the sum of its individual parts. And although convolution-based models lack the biological detail of conductance-based models, they provide a more straightforward and interpretable framework for understanding these system-level dynamics of neural populations. It is also possible to see convolution-based models with conductance-based elements. It all depends on the level of mathematical tractability and biological plausibility that you're looking for. And so what type of processes and dynamics you're trying to study and investigate. Convolution neural mass models are composed of two key operators. So they have a rate to potential operator that describes the dynamics between synapses and dendritic trees. And this is usually in the form of what is called an impulse response, because it has been shown that the convolution of an incoming spike rate with an impulse response will adequately reproduce a postsynaptic potential that is responding to a presynaptic firing. And this convolution can be expressed as a second order differential equations that you see here. And the two main parameters of that equation is the maximum amplitude of that postsynaptic potential and the tau e, which is the rate constant and determines the steepness of the slope, so how fast the signal will propagate, will be transmitted. And then there's also a potential to rate operator, which is represented by a nonlinear function and generally a sigmoid, and this transforms the average membrane potential of the population into the average rate of action potentials fired by the neuron. This is also known as the wave to pulse conversion, so it does the inverse process of the rate to potential operator. And I just wanted to mention that the sigmoid is not derived from a biophysical model, but it's rather seen as a physiologically consistent choice. And the parameters here are the maximum possible firing rate, There's also the value at which 50% of the firing rate is reached, at which voltage, and then R will determine the steepness of the slope. And so by combining those two operators, we are then able to represent the coarse grain activity of each population in the model. So each population, neural population represented in the model will be of the form of nonlinear second-order differential equations for convolution-based model. And this can be either deterministic or stochastic, depending on the external input that is introduced to the model. So now I'll be going over the 3 different neural mass models that we will also be investigating in the hands-on session right after. So the Jansen-Rit model is a cortical model composed of 3 populations, and it is used to reproduce EEG data. We then have the reduced Wong-Wang model that was developed by Deco et al., which can be either composed of 1 or 2 population, and the output of that model is usually transformed to then replicate fMRI data when it is integrated into a network. And then we have the Robinson-Rennie-Wright model, which is a cortical thalamic model. And this was originally a neural field model, but when spatial uniformity is assumed, it can be likened to a neural mass model. Jansen-Rit model is one of the first neural mass models that was developed to reproduce so EEG and alpha activity specifically, and is described to the macroscopic electrophysiological activity within a cortical column. So it represents a patch of the cortex, and it was popularized in 1995, and many future models were derived from it, and it is still widely used. So I mentioned already it has 3 neural populations, pyramidal cells, excitatory and inhibitory interneurons. And the fact that pyramidal cells are separated from excitatory interneurons is due to the fact that the neural activity captured by EEG is mainly from pyramidal cells. So here we have the detailed schematic of the model. And as you can see, we have the 2 operators that I mentioned previously. So the nonlinear sigmoid operator followed by the impulse response for each populations. And the C represents the connectivity between the different neural populations. So I'm not going to go into the details of the equations, but I just wanted for you to visualize the fact that each of them are composed of a nonlinear second-order differential equations that are decomposed into 2 first-order differential equations. And I also wanted to mention that in this model, there's a different excitatory postsynaptic potential and inhibitory postsynaptic potential, which are defined by big A, big B for the maximum amplitude, and small a, small b for the rate constant, and it is different for the PSP and the IPSP. So the output of the model which is represented here by the yellow cross, corresponds to Y1 minus Y2, and this is so the output of those pyramidal cells, as you can see here. And when the Welsh method is applied, we get then the power spectrum, and we can see this characteristic peak in alpha, so around 10 hertz that I was mentioning, that is usually seen during resting state. So here I just wanted to present an example of how this model can be studied and used. So in this case, we performed a steady state analysis to investigate how the dynamics of the system changes as a function of this excitatory inhibitory connections, which is actually altered in certain diseases. And so, for example, in this case, so we have on the y-axis here what is called these fixed points. So when so this is part of the steady state analysis, and essentially what this is telling us is that when the connectivity parameters are within the part where we get this red curve, we get an oscillations, and the frequency of which tends to decrease as you go along, as this value increases. And when the connectivity parameters are within this blue region, there is no activity that is seen. And so this really shows that there are certain conditions for which you will get brain activity or not within these neural population, and that a certain balance exists between these 2 parameters. And in this second figure, so here we have actually lowered the input that is given to the system, and we can see how the shape of this curve changes and how you have less possibilities of getting oscillations. So this is kind of an analysis that is pretty useful because you can gain insight on how these different large populations communicate with each other pretty easily and visualize it. We then have the reduced Wong-Wang model, which refers to the model that was developed by Deco et al. And first presented as a single population in 2013 and two populations in 2014, which is what I'm showing here. So this model is actually derived from the Wong-Wang model, which applied what is called a dynamic mean field approach to a conductance-based spiking local attractor model. So this model is actually derived from a conductance-based models, and so has a synaptic gating variable, which was not seen previously. So in this model, you have input current that is transformed into firing rate, so also using a sigmoid, and then the firing rate will be used to infer what is the synaptic gating variable. So, and the synaptic gating variable is then the output of this system. And there's the same is applied to the inhibitory. So the same is applied to the excitatory and inhibitory population. And so here is said that the excitatory actually represents an MDA, because the synaptic gating variable of NMDA receptors has a much longer decay time constant than AMPA receptors. The dynamics of the NMDA gating variable dominates the time evolution of the system. So this is why AMPA receptors here are not represented, because the synaptic variable instantaneously reaches its steady state. So here, as you can see, I have set these parameters here equal to 0. So this is the case when you have an isolated node, so just one node of the simulation here. And with the specific parameter values used, the mean spiking activity of the excitatory pool for then this isolated local network is around 3 Hertz. And then, as I mentioned previously, when these nodes are not isolated and different nodes are assembled together, this model is used in conjunction with the balloon-Wien-Kessel equation to transform the simulated excitatory synaptic activity into a bold signal and be able to replicate fMRI data. So yeah. We then have the Robinson-Rennie-Wright model. So this model is a cortico-thalamic model where you have excitatory and inhibitory cortical neurons and two thalamic population, the reticular nucleus and the relay nuclei. And so this physiologically based continuum model of cortico-thalamic dynamics can reproduce and many features of EEG. And so this model, actually, the equations are very similar to the Jansen-Rit. So you can see the sigmoid operators and as well so the sigmoid operator, and it's also the impulse response. But it also has, as you can see here, a phi E. So this is due to this damped wave equation operator that is added to it. So here you have the same thing where the mean voltage becomes firing rate, but the firing rate can also be propagated and be damped over time as it goes along the axonal tree. And so that's why you have this additional phi A here signal, and then this is converted into voltage instead. However, we can make the approximation that actually firing rate is equal to phi A when you assume that there is no propagation. However, so there is only the phi E that is left, because propagation delay and so long axonal ranges are solely preserved for the cortical excitatory population, as it is actually the only population that is large enough with distant connections for wave propagation to have a significant effect. So that is why this is the only case where you have here the damped wave propagation that is applied. And the other difference with the Jansen-Rit model that you can see in addition to the fact that it's different population represented and different parameters, you also have this delay term from when the information is passing from the cortex to the thalamus and then the thalamus to the cortex. And so the output of this model is this phi, the output of the cortical population, and the signal load is what you see. And the same here as we apply the Welsh method and have the, we can see, so then again, this alpha peak around 10 hertz. So what is interesting with this model, and that I wanted to mention, is that a lot of analysis has been done with the linearized version. So they were actually able to reduce the number of dimensions and connection to succinctly describe the dynamics in the 3D space that represents the different main interactions. So you have here the cortical loop with the GE, GEI. You then have a corticothalamic corticothalamic loop, so through the GSRE here, that goes through the reticular nuclei to the relay nuclei, and then just from the pyramidal to the relay nuclei, and then back to pyramidal. And then the final interaction is this intrathalamic loop here. And so they were basically able to simplify all the connections and represent in an XYZ space representing corticothalamic, and thalamic stability. And what's very useful here is that, for example, here you can see as you increase the corticothalamic interactions, you tend to get alpha, as when this parameter will decrease, you will get delta and theta. So it's an easy way to visualize and see how the dynamics of the system change based on these different loops and interactions between the cortex and the thalamus and so on. So now we have seen different neural masses and what is possible. And now I'm going to go over how these are, because they each kind of represent parts of the brain, but not the whole brain activity. And this is where the Connectome neural mass model simulation comes in. So, because neural mass models are point mass models, so they evolve in time, but not in space. So to simulate whole brain activity using those neural mass models, they are coupled according to a weighted connectivity matrix that represents the strength of the anatomical connections. That is known as the connectome. And it is often estimated with diffusion weighted MRI data, so tractography that you can see here. And then each node of that corresponds to a neural mass models, and that depicts a brain region that is determined by the parcellation that you will be using. And this collectively then forms an integrated brain network model. And this model can then be tuned and fitted. So your simulated signal can be fitted then with your functional data, so the fMRI, EEG, or MEG, to tune the parameters. So one important parameter, for example, is a global scale long-range coupling factor that will kind of rescale the structural connectivity that you got from the empirical data, and change so the strength of the interactions between the different nodes. So I'm going to end the presentation by going over a few examples where connectome-based neural mass modeling was used. So in the first instance here, it was used by Momi et al. To investigate the effects of lesions in the structural connectivity, and more specifically, how does the signal propagation after a stimulation changes when there are lesions in the structural connectivity. And they did that using a whole brain connectome model. And so for instance, here in the first instance, they were looking at how, depending on when the lesion is made, the signal will either propagate or not. So this kind of investigates the speed of the signal propagation dynamics. And then in the figure below here, you can see how it is completely when the structural connectome is lesioned, the neural activity of the brain is completely altered, and we do not get as much activity. So this really shows how the lesions in the structural connectome can affect the neural activity of the brain and the brain dynamics, all this using a whole brain connectome neural mass model, whole brain model. And finally, in addition to using it for research, it can be used in a more of a clinical application as well. So a few examples, there will be actually presentation later within this education going over these examples more specifically. But I just wanted to mention that, for example, it's been used to study Alzheimer's disease, and using a platform called the Virtual Brain, TBB. So it's a neuroinformatics platform for full brain network simulations. And so it provides a framework to simulate connectome-based neural mass models. And what they did here is they used a Jansen-Rit model, but altered it, modified it to be able to integrate the differences that can be seen with the deceased an Alzheimer's deceased brains. For example, how represents the alteration that can be observed to the amyloid plaques versus a healthy brain using whole brain modeling. And so this is work that's been done by Stefanovsky et al. It can also be used to study seizure genesis and propagation. And this was also done. So this work by Dolomaj et al. Was also done in the virtual brain, and they use what is called an epileptic model. And so this can use, for example, data from patients who experience seizures and can be used like for pre-surgical evaluation. So in conjunction to that, this type of whole brain modeling can be used to help neurosurgical procedures before they're done and so on. And so they are able, since you have a model fitting part, you can create those personalized brain models and really study for the patient how seizures, how the seizure propagates in the whole brain and so on when it is modeled. So in conclusion, we have first seen how EEG and fMRI record whole brain activity and how modeling can be used to understand so the underlying brain dynamics of the phenomenon observed with those modalities. Neuromass model, they describe the coarse-grained activity of neural populations, and it has two main operators, a rate to potential and a potential to rate operator for convolution-based neuromass models. We saw three different models: the Jansen-Rit, which is a cortical model. We then have the reduced Wong-Wang model that is used to replicate fMRI data. And then we have the Robinson-Rennie-Wright model, which is a cortical-thalamic model. So all these three models will be investigated in the hands-on session. Neuromass models, they represent the local dynamics and form the nodes of this whole brain model, and the network connections are inferred by the structural connectivity and tractography. And they can be used in multiple ways to study the effects of lesions, stimulation, evolution of different diseases, such as Alzheimer's disease and epilepsy, which you will hear more about in the other presentations. So I wanted to end this presentation by thanking the whole lab, and especially my supervisor, Dr. John Griffiths, and Dr. Davide Momi for organizing all this educational course and inviting me to do this talk. Thank you for listening.